This is going to be chapter 6 review. There's a lot to go over, but should be able to get through everything. Hopefully in a timely manner. I got everything written down. So let's get started. The first things we talked about in chapter 6 were the microscopes. Of course, we're using the light microscope. The light microscope is the one we're using, but we also went in depth on the electron microscopes. The two microscopes that we looked at or that we learned about were the transmission electron microscope, which is used to look inside things, and the scanning, which is used to look at surfaces. So just remember, like, scanning is just to scan the surface, kind of. Just you scanning, you're not looking inside it. And transmission is, just going inside and looking what's inside. That is it about microscopes. The next thing we looked at were something about eukaryotic cells versus prokaryotic. Of course, we know that eukaryotic cells um, sorry about that. We know eukaryotic cells has membrane-bound organelles. We learned that in the first section. Um, the only thing that we need to know is that the space where DNA is stored in a prokaryotic cell is called a nucleoid. That's the space that it's actually located in. That's the space. It's not a membrane-bound place, but it's just the space it's called. And then for DNA, it's stored in the nucleus. Another thing we went over was how surface area affects cells. For example, if this was a cell 1 and we had multiple cells on this side, this is multiple, more than one cell. Food distribution um, wouldn't be as effective. The food would go in here, and before it could even reach maybe this corner uh, for the cell, everything would already be eaten up by most of the um, stuff on this side. So what they do is they break down the cell into multiple cells, or not multiple cells. They break it down into a smaller surface area, so that some food would go into that cell, some food would go into that cell, some food would go into that cell, and it all just it works out better. That's all you have to know about surface area. It's just better for food distribution of the surface area of a cell. The next thing we went a little bit into, not in depth, we'll go into depth in chapter 7, was the plasma membrane. It's made out of these hydrophobic heads. Oh, sorry, hydrophilic heads. My fault. Uh, they're phosphate. It's a phosphate group, a phosphate hydrophilic head. And then you have these hydrophobic tails right here. So it creates this hydrophobic tail and a hydrophilic head. And why this is important is because this area right here would be a liquid, so it'd be hydrophilic. So this area right here would be hydrophilic. The side inside the cell would be hydrophilic. So it needs a hydrophilic outside, and the hydrophobic inside will help with other things like selective permeability and um, and just helping keep the cell intact. Okay. So now we move on to where genetic information is stored and stuff like that. Um, we talk about the nucleus of eukaryotic cells and the parts of it and what they do. So this is our nucleus. The whole thing is the nucleus. Um, the parts that make up the nucleus is pretty simple. You have your nuclear envelope, and why it's called an envelope is because it has an outer membrane and an inner membrane. So it creates this hollow area where if you have your outer membrane right here and you have your inner membrane right here, you have this space in between, which will go over kind of helps with, it's kind of part of the endomembrane system, and it helps with creation of proteins and stuff like that. We can talk about the what's on the outer membrane for a second. We have ribosomes on the outer membrane. There's also two other locations in the cell that ribosomes are located, and nuclear pores. Nuclear pores help, like it's like a security measure. It helps know what things go in and out. It helps with things going in and out of the cell and protects things that's not supposed to go in it, not go in it. Um, we have chromatin. Chromatin is kind of free floating around. We'll go over probably in another chapter how it forms as a chromosome when the cell is splitting. We'll talk about that in another chapter, but for right now, just know that chromatin is practically chromatin is basically DNA wrapped around proteins called histones. So you have your um, histone proteins, and you have your DNA, which will be wrapped around it, and then there'll be multiple of those. You have your protein wrapped around it, and that would be chromatin. So chromatin is basically DNA and proteins called histones, and it's just tightly wrapped. Again, chromosomes are chromatin wrapped around, so a bunch of these chromatin wrapped around each other would be make a chromosome. So that's another step that they talked about. Next and last thing is the nucleolus. Um, the nucleolus, uh, the nucleolus is where the ribosomes are made, or synthesized. Um, it also holds the R, oops, rRNA, and also ribosomal proteins are located in here. And that is pretty much all you have to know about the nucleus. So we got our nuclear envelope, we got the nuclear pores, nucleolus, and chromatin, which make up the main part of the nucleus, and then we have ribosomes that are attached to it, the outer membrane. And we'll also talk about how the outer membrane of the nucleus connects to the rough ER. Now we can have fun and talk about the endomembrane system, which is it's a long process, but um, pretty simple to understand. It's basically just the making of membrane proteins and secreted proteins and lipids, as well as lysosomes. So we'll go over the steps real quick. As you can see here, we have our rough ER, and partly this part here would be the outer membrane of the nucleus. These little guys here would be ribosomes, and this whole thing right here is called the rough ER, or the full name is rough endoplasmic reticulum. I don't know how to spell it. Did I say here? No, I didn't. I think it's like that. I might have it wrong, but um, so basically the whole part we have to put it in steps. Um, put it in steps so it's easier to understand. First of all, what happens is we also have the smoothie which I'll just draw arrows from it because I can't really smoothie R. The smoothie R, I'll do this actually. The smoothie R has two jobs. We'll talk about the smoothie R. The smoothie R synthesizes lipids because it does not have ribosomes. It also detoxifies toxins that may happen to come in the cell. It would go to the rough ER, the smooth ER, and that would detoxify them. But another big thing here is the synthesis. The smooth ER synthesizes lipids, and the rough ER synthesizes proteins. Those proteins and lipids are sent to a transitional ER somewhere on in the rough endoplasmic reticulum. And that those rough endoplasmic reticulums and would take the proteins, and from the smoothie R would take the lipids and put it out in a vessel or a vacuole. So a vessel is just it's phospholipids. It's a bilayer phospholipids holding those proteins and those lipids and transporting them. And where they're going to transport it to is the cis side, so the cis Golgi apparatus. So right here, this is going to be our Golgi apparatus. Oh, another word I don't know how to spell. Apparatus, I think. The Golgi apparatus right here. And you have two sides. You have the cis side, which is the entering. So entering. And the trans side, which is the exiting. Um, the whole purpose of the Golgi apparatus is to um, receive, sort, and ship these proteins and lipids. Now, what else could happen in the Golgi apparatus while the lipids and proteins are going through the Golgi apparatus is it can get so while it's getting modified, it can turn into a glycolipid or a glycoprotein as well. So that's something to keep in mind for Golgi apparatus. A glycolipid and a glycoprotein are basically carbohydrates added to um, the proteins and the lipids. And that's just another thing the Golgi apparatus does. Now, once we have the lipids and the proteins inside the Golgi apparatus, those have to go somewhere. And this is where you have three things that could happen. One of the three things is, again, it could have secreted proteins needed to take in a vessel, and it can secrete them outside the cell. So it could secrete proteins such as insulin. Insulin is one. Another thing it can do is it can have membrane proteins that need to be combined with the plasma membrane. So the vesicle would join in with the plasma membrane as well as the protein. So it would all become just, it would just become part of the plasma membrane. So that's the second one. Um, Proteins and vesicle, ves part become part of the membrane, and that'd be two. And the third and last thing it would do is the lysosome right here. The lysosome, the lysosome has hydrolytic enzymes, which are also proteins, or digestive proteins that um, they digest things in the cell. So lysosomes will digest things inside the cell. They can also digest the mitochondria, and we'll talk about that more a little later when we get into peroxisomes. But lysosomes will go around and digest, okay, of course, food. They'll digest anything that needs to be recycled and stuff like that. It would be done by the lysosome. So those are the three. The lysosome is one, what, and the vessel will become the lysosome. Once the proteins are put inside the vessel and it leaves the Golgi apparatus, it becomes the lysosome. So the vessel and the stuff inside, which is the hydrolytic enzymes, become the lysosome. So that is the entire endomembrane system. If they ask you what parts make up the endomembrane system, the parts would be um, the outer membrane of the nucleus, the rough and smooth ER, so the rough ER and smooth ER, um, the Golgi apparatus, transport vessels, the lysosome, and don't forget the plasma membrane is also one. The plasma membrane is part of this process. So that would be it for endomembrane systems. Let's talk more about vacuoles or vessels. You can have there's three main types of vacuoles that we'll be talking about. A food vacuole, for example, is just when food is entering the cell. For example, if this is your cell membrane, we talked about phagocytosis. So when food is entering the cell, it's called phagocytosis. And you have your food here, and when it enters the cell, then you have your food, but it'll be encased in a plasma membrane. And this would be the food vacuole right here. It's called phagocytosis. This process right here is called phagocytosis. When its food is entering the cell, it's called phagocytosis. Another main vacuole that we'll be talking about is the central vacuole, which is usually in mature plants or plants. Um, Only in plants. The main purpose of the central vacuole is to store food and sugars and stuff, and it removes toxins. So really important in plant cells. The contractile vacuole pumps extra water out of the cell so that um, whatever has a contractile vacuole does not like just burst, does not lyse in water. Um, it can survive in water. I'm pretty sure it's the paramecium. The paramecium has a contractile vacuole that pumps the water out of the cell once it's full, so it does not burst and does not it can survive in water. So that's another important vacuole. Can move on from that. Now we'll be talking about the mitochondria and the chloroplast. Um, Pretty. straightforward. We just talk about what it is and kind of what it does. So the part of there's two types of membranes in a mitochondria. That would be the outer membrane and the inner membrane. Now the outer membrane is relatively smooth. It just kind of protects the inside, and the inner membrane has foldings. These foldings are called cristae, and why they fold is to, of course, increase surface area, which will, of course, come in handy for cellular respiration. The cristae is the folding, so we can call it the inner membrane foldings. Um, we went over endosymbiosis. Endosymbiosis is the theory of what might have happened to the mitochondrial chloroplast, where the mitochondria was and, absorbed by the eukaryotic cell. So that's probably where they got the outer membrane from, and have the inner membrane as the actual might be bacteria. Um, Other than that, we know what is inside of the mitochondria is the matrix. The matrix is the space inside, and this will come in handy when you have to know the difference between a chloroplast and the inside of a chloroplast and the inside of a mitochondria. So the matrix is anything, any space inside the inner membrane. Of course, in the inner membrane, we have the mitochondria DNA and ribosomes as well. So ribosomes do create protein, so that means inside the mitochondria, it does produce proteins, and it also has its own set of DNA. So it's really important to know that mitochondria has its own set of DNA. The next thing we have is the chloroplast. Um, don't think we really talked much about it in class, but it kind of has three membranes. It has an outer membrane, which is smooth. It has an inner membrane, which is also smooth. It's not like foldings like the mitochondria. But now there's three new terms that we have to learn here. The thylakoid is one of these circles. I like to call them like cookies. I like to call them like cookies. They're sacs. Uh, they're sacs that are used to turn chemical energy, or sorry, light energy, um, into food or sugars. Multiple thylakoids or multiple of these cookies, I like to call them, can be stacked like this right here. And one stack will be called a granum. And I like to call them like a stack of cookies. So thylakoids will be one cookie, while a granum will be more than one cookie or just a stack of cookies. The space inside of the inner membrane, again, it's not called the matrix, it's called the stroma, so you can't get them messed up. Uh, Stroma is the space inside the inner membrane. Again, so space within inner membrane. So yeah, now the whole point of um, uh, these two organelles is that the mitochondria basically carries out processes to produce ATP for the cell. And we have to remember that plant cells also have mitochondria. They don't just have a chloroplast. Plant cells have both the mitochondria and the chloroplast. And so mitochondria carrying out cellular respiration and the chloroplast carrying out photosynthesis um, will really help both type of cells. Um, so that would be it for mitochondria and chloroplast. Next thing we'll talk about peroxisomes. So peroxisomes we talked about. I like to think about they give us two examples here. But they break down toxins, but they also are used for different reasons. The reason we talked about in class was they break down the mitochondria. So when you have your lysosome here with your hydrolytic enzymes in it, and you have your mitochondria, for example, just gonna draw it like that, and you have your mitochondria, so this would be your mitochondria, and this would be your lysosome. The mitochondria, when it gets broken down by the lysosomes, produce toxins, and that's where peroxisome comes in place. Um, Peroxisome takes hydrogens and adds oxygens to them to make them hydrogen peroxide. We'll talk about more about that. Peroxisome. So it basically takes hydrogens from toxins. So when all these three come together, there'll be a membrane around all of them. While the lysosomes are breaking down the mitochondria, the peroxisome is taking those toxins and turns them into H2O2, which is hydrogen peroxide. However, there's a problem. Hydrogen peroxide is also toxic. However, um, the cell is very clever and knows that it can break down hydrogen peroxide. It can break down hydrogen peroxide into water. So it's actually better to take that toxin than the toxins from the mitochondria, which could be really bad. Another reason I found in the book is they could be used to break down fatty acids and send them to the mitochondria when the cell needs ATP. That's another thing a peroxisome does, um, and that's also really helpful. So now that we're done talking about peroxisomes, we have to talk about um, the next part of the cell, which is the cytoskeleton. Now, there's three parts to the cytoskeleton, or I should say three things in the cytoskeleton that make up the cytoskeleton. And all three things you have to know, all three things give the cell structure. All three things give the cell structure, shape, and support. We call them the three S's. All cells get structure, shape, and support from all three parts of the cytoskeleton. However, there is movement involved in two of them that we'll go over right now. The first thing we'll look at is microtubules. Microtubules are just hollow rods found in all tubules are just basically hollow rods found in all eukaryotic cells. They kind of look something like this, and the inside here is hollow. Um, and they mainly do they also do structure, shape, and support. So you have to know that microtubules do structure, shape, and support, but they also do movement. And they have two types of movement. And they have movement in the cell and movement of the cell. So if you look at movement in the cell, the main thing microtubules does is it has a wide network of microtubules throughout the cell, and things move using these microtubules. It's like a train system. We have motor proteins, which basically use ATP. Um, so they use ATP and they basically walk. They walk on these microtubules attached to a receptor on a vessel or receptor proteins on anything, for example, a mitochondria or anything that needs to move. And move, the vessel will be moved through the motor protein across the microtubules. So the motor protein will be walking this way and it will keep walking until it needs to get to its destination. That is it for the movement in cell of a microtubule. Now we talk the movement of the cell. Um, Movement of the cell, there's two types of movement of the cell through microtubules. Microtubules can create flagella, which is also another example found in sperm cells. It's just, it has like snake-like movement and it has only like one tail, so it's like a one flagella. Cilia, however, are multiple hair-like structures like this found on the surface of the cell that also are used to move and can also stick to objects. So here I made a list of what they do. They both help move the cell. They both help move the cell and are in a 9-2 formation. What that means is basically 9-2 formation just means they have uh, two microtubules in the middle. So that's where we get the two from, the two microtubules in the middle. And then we have nine sets. We have nine sets of two, nine sets of two microtubules. So um, we'll have that is one set of two. So that'd be one. That'd be another one. That'd be another one. That'd be another one. One, two, three, four, five, six, seven, eight. Now I drew them kind of too far apart, but this is the 9-2 formation it's talking about. And what they have is they have proteins in between them, connecting them. They have proteins like, I mean, I know I drew them kind of close, but they basically have like proteins connecting these that help them move. So I talked about it here. Uses motor proteins called dyneins to work. So these would be considered the dyneins, and they'll walk across these microtubules. So these are microtubules. So those pink things, the formation is formed by microtubules. Um, so the flagella and cilia are made out of a 9-2 formation of microtubules, and they use basically motor proteins to move. They also connect to the basal body. So the basal body is basically inside the cellular membrane. For example, if this is the cellular membrane, the basal body would be down here, and the flagella would connect to that. For example, like this. So the flagella is kind of held in place by this basal body, which is not a 9-2 formation. It's a 9-0 formation, which is just nine sets of three microtubules. So that'd be the same thing as a centrosome and a centriole. So um, we need to talk about the centrosome. A centrosome just consists of two centrioles, which are made out of microtubules. Their main purpose for is during the cell splitting, the centrioles will help move the chromosomes and stuff like that. Uh, they're made out of 27 microtubules in a 9-0 formation, so basically 9 times 3, because they have 9 sets of 3. So that's where I said the basal body has the same formation as a centriole. And the centriole, for example, the centriole would be these two guys. Sorry about that. Would be these two guys in the cell right here. Um, and they're made out of microtubules, and they help move chromosomes around during cell splitting. And they're also just like the main hub of all microtubules. think I hit everything. Yep, I think I did. All right. Next thing we talk about is microfilaments. Um, Microfilaments also three S's. Oops, three S's. Structure, shape, and support. Um, it's basically a cable made out of actin proteins. So they're made out of actin proteins. Um, we looked at an example, a structural example of structure, shape, and support. A structural example for this reason would hold the membrane in the for the muscle for the stomach for the sorry for the intestine cells to collect food. They need a lot of surface area. So a better way to do that is to create more surface area, which is have your plasma membrane kind of do this thing where it allows for more surface area, the foldings. One folding is called a microvillus. So one fold will be a microvillus, and multiple folds will be a microvilli. And that's in the intestine proteins, sorry, intestine cells that help digest food. Not digest food, sorry. That helps collect um, the resources from food. So that is really important to us. Um, the actin proteins kind of help hold the microvilli by basically just creating like this structure here, multiple structures just holding these together, like a bridge kind of holding this up like this. So these lines and folding, oh sorry, these lines would be holding those together, and there's a lot of them. That is a structural example of a microfilament. Um, Now we move on to movement of microfilaments. There's three types. There's movement of muscle contractions. For example, we have muscle contraction, which is basically just myosin heads walk on the actin to contract. So here would be a picture of that. We have the myosin heads, then the myosofilaments holding the heads, and the actin filaments. And what would happen is these heads would walk just like kind of like motor proteins, and they'll contract the actin filaments together. So these arrows are showing the contraction happening. And once these come together, and you wanna uh, uncontract, or when you don't want to contract anymore, then the heads walk the other way, and your actin filaments would go this way, causing you to um, not be contracted anymore. Another another movement is pseudophodia. Pseudophodia in amoeba cells. Um, the actin and myosin interact to pull itself forward. So while these actin and myosin filaments are doing their job to like kind of extend, it's also taking this and it's taking the back of the cell and it's making it smaller. It's bringing it forward. These arrows are showing the direction of where the cell is going. So if the cell wants to go this way, it will expand the cell and it will de-expand from this side, so it'll make it less. It's called, you can call it false feet, but the main thing is called pseudophodia or pseudopodia. Sorry, pseudopodia. Um, the last one is cytoplasmic streaming. Cytoplasmic streaming is in plant cells, and it's the flow of cytoplasm that speeds up material distribution. Um, you can also think about it as chloroplast in plant cells. If you have a chloroplast right here, for example, like that, and it's kind of staying there, it's not doing anything, then and the sunlight, for example, is hitting this way, then the plant cell is getting no sugar, no energy. So what it does is the plant has this cytoplasmic stream to move all chloroplasts around to so that they all get sunlight. And it's a pretty clever way for all the chloroplasts to get sunlight and turn it into sugar and energy for the plant cell. And it also uses actin and myosin filaments to do that. And that is it for microfilaments. The next and last one of the cytoskeleton is intermediate filament, um, and this is pretty straightforward. It just anchors. All it does, it does is structure, shape, and support, the three S's. It anchors things in place. For example, the nucleus. Intermediate filaments help hold this nucleus together in its place because if the nucleus is moving around, that's not good. If the rough ER or Golgi apparatus is moving around, that's not good. You kind of want to keep it in place. Whatever moves, it does not deal with it. It's only used for anchoring. It's only used for anchoring. So all it does is hold things in place for it to not move around. And that's it for the cytoskeleton. Now for the last part is uh, extracellular components for the plants and animals. Um, the first thing we will talk about is a cell wall, usually found in plants. Um, Usually they're found in plants. Most prokaryotes have them, so like bacteria and stuff like that. Fungi have them, and some protists. Um, and all it does is protects the cell. When water goes in the cell, it protects it from exploding. It protects it from other toxins and stuff like that. It just protects the cell in general. It's a good protection layer. As for the animal cells don't have a cell wall, but they do have something called an extracellular matrix. Um, and it looks complex, but it's really not that much. Um, you have your collagen, which is a glycoprotein, which is probably the main thing holding everything in place. It's a glycoprotein. Um, you have your polysaccharide. You have your proteoglycan complex, which is just a complex of a core protein and a carbohydrate. And we'll talk about more of that. We have fibronectin and we have integrins. And as you can see here, we have a phospholipid bilayer I kind of drew out to describe where everything is. So this is where my arrow is going is outside the cell. So this is outside. This is why it's called extracellular matrix. It's outside the cell. So we have a extracellular matrix, which we can start off by talking about the integrins. So the integrin proteins are inside of the plasma membrane, and what it does is it connects to the fibronectin. And basically, what the fibronectin does, it's like a communication between the integrins and the collagen and what's going outside the basically tells the cell what what is going on outside. Can I come in the cell? If something wants to come in the cell, it basically tells them, oh, we have this. Can it come in? And it'll transfer the data. So we have a collagen, which is a glycoprotein. We have a polysaccharide. Well, connected to the glycoprotein, we have a polysaccharide um, molecule. Sorry, not molecule. We have a polysaccharide like string, kind of. And connected to that polysaccharide, we have a proteoglycan complex, which is just made out of two components, which is a core protein and like carbohydrate hairs coming out of them. And the whole purpose of this is just to protect the animal cell as well from a lot of things. It protects the animal cell. And that would be it for the extracellular part.
Now, as for the last part of chapter 6, we talk about cell junctions, and the main one is in plants we have plasmodesmata. Now, for animal cells, they also have the exact same thing, but it has a different name. It's called gap junctions. Gap junctions here. Oh my gosh. We have gap junctions over here, but for for plants specifically, it's called plasmodesmata, and it's just basically cell communication and sharing of the material. So right here we can see we have two cells, and we have plasmodesmata where if cell 1 needs something, cell 2 can give it to them. Or if they just need info, like, hey, what you doing? They can just share info. As for animal cells, we have three types of junctions. We have a tight junction. This prevents leaks of resources between cells. So, for example, this is again the intestine cell, um, where if stuff were to go down the cell right here, if the tight junction wasn't stopping it, usually it'll go here and it'll stop because it cannot go any further. That's what a tight junction does. But just say if we didn't have a tight junction, if things went all the way down the cell, we can get infections and stuff like that. So tight junctions is really important for, um, preventing liquid and preventing food and stuff like that to going down the cells, so between the cells. Another type of um, cell junction we have here is a desmosome. It holds the cells together like a solder. Like if you were soldering something, we have here intermediate filaments. These hair-like structures are intermediate filaments. Um, and the desmosome basically just holds the cell together. It solders the cell together so they don't move around, and we can have tissue and stuff like that. Like muscle tears. When you have muscle tears, it probably these desmosomes will probably break, and that's how muscle tears happen. The cell will kind of break the desmosomes, and you'll have muscle tears. And finally, for the last cell junction, in animal cells, you have a gap junction over here. It does the exact same thing as a plasmodesmata. It shares information and resources with the other cell. So if this was cell 1 and this was cell 2, these plasmodesmata share information and also um, food and stuff. For example, information would be in the heart. Um, the heart has to pump. All the cells have to pump together in a group. So the gap junctions will tell the cell, well, each cell, hey, I'm gonna pump now. I'm gonna pump now. If they were not pumping properly, you would not be alive. The gap junctions are really important for communication and sharing of material. So that would be all for chapter 6. We went over um pretty much everything over chapter 6. And there's two more chapters for the exam, which I'll try to get up and posted. So stick through it. Um, if you guys need any help, just let me know. All right, have a good day.